Assalamualaikum and good day to everyone. My name is Siti Zulaikha binti Zainal Abidin, and today I'll be presenting my final year project research entitled The Effect of Eggshell as a Growing Medium Treatment on Plant Growth Performance of Red Spinach. In this table of content, there are 5 chapters that will be discussed. The first one is the introduction, second is literature review, third is methodology, fourth is results and discussion, and the last one is the conclusion and recommendation. In this background of the study, it is about the growth of red spinach inside soilless medium growth containing cocopeat, in which the system used is known as hydropipe. The spinach growth is the same as like other plants where it requires both macro and micronutrients. The main source of fertilizer to this plant is coming from the eggshell, which act as a natural source of fertilizer. Plants require sufficient amount of nutrients and mineral from soil in order to have a good growth yield. However, using soil as a growing medium could have limitations in terms of the nutrient content. Application of fertilizer to boost the nutrient content inside the soil bring more drawbacks than positivity. Fertilizer could leach and cause pollution to the groundwater. 50% to 70% of fertilizer potassium are lost to the atmosphere and cannot be absorbed by the plants. This is not only cause resource losses, but also environmental pollution. Besides, plantation of vegetable crops in soil require continuous physical work, especially for watering and supplying nutrient process. Limitation of the plantation area somehow limits customer demand and the production yield are too low. For significance of the study, the first one is cocopeat has good physical qualities where it is high in porosity. Ability of retaining high water content will make sure high wettability of mixture. Plants have continuous supply of water even when the watering process is reduced. Second, eggshells are the safest source of calcium to the plant. It promotes biological fixation of nitrogen gas in acid soil and increases the net organic nitrogen mineralization. Besides, it elevates the pH level of highly acidic soil, making it favorable for plantation. Third, modern agriculture system is introduced to meet the food demands. Soilless growing system is a way to obtain high yield plant in a limited area without disturb its growth yield. Scope and limitation of study is divided into 4, which is the first one is the study focuses on the growth of red spinach using cocopeat as a soilless growing medium under different percentage of eggshells. Second one is the treatment was done in 3 different percent of eggshell, which is the first one 0%, which is 100% cocopeat. Second is 50% cocopeat, 50% eggshell. Third one is the 80% cocopeat, 20% eggshell, with each treatment have 4 replication and each replication has 2 individuals. Third is the growth of the plant was measured in terms of height, number of leaves, and also the fresh weight. Fourth is the chemical analysis used to study the chemical composition of eggshells and cocopeat and how the growth of red spinach is influenced by this treatment. The objectives of this study is divided into 2, which is the first one is to determine the chemical characterization of cocopeat and eggshells as soilless growing medium using chemical analysis. The second one is to investigate the suitable growing medium ratio on plant growth parameter of height, number of leaves, and also the fresh weight. Chapter 2 is the literature review. Hydropipe system is applied as a soilless growing medium system in this study. When compared to the traditional soil farming, this technique gives a proportional larger return and better quality crops. This system allows planters to manage nutrient, oxygen, and water needed for optimum plant growth. Application of cocopeat in this system improves the efficiency of water consumption and fertilizer to its high water holding and cation exchange capacity. With this combination, plantation process can be done a lot easier and production costs will be reduced. As plants require enough macro and micronutrients, the use of eggshell was applied in the study as a natural source of calcium. Besides, eggshell have been found to be the most effective alternative to cultures. With high calcium content and ability to reduce soil acidity, the use of eggshell in this study is believed to improve crop yield. Chapter 3 is the methodology. In methodology, the first step is sample preparation. Red spinach seed were first germinated in trays for 7 days and placed in the propagation area. During this germination process, hydropipe system is built up. Step 2 is experimental setup. Hydropipe uses PVC pipe where it has holes on the surface for medium pots to be placed. Each pot then has holes on the bottom which allow plant's root to absorb the nutrient solution. A small pipe was inserted on the one end allowing the entrance of the nutrient solution. Step 3 is experimental design. After 7 days, red spinach is planted in 3 different treatments. The first one is 100% coco peat, second is 50% coco peat, 50% eggshells, and the third one is 80% coco peat and 20% eggshells. This treatment is replicated into 4 and each replication has 2 individuals. Step 4 is plant growth analysis. Plant height and number of leaves is recorded after 5 days in 35 days growing period. After 35 days, the plant is taken out from the pots and wait for its fresh weight. Step 5 is chemical analysis. During this growing process, chemical characterization of coco peat and eggshell is analyzed using chemical analysis known as ICP-OES and also FTIR. And the last step is step 6 is the statistical analysis. In this statistical analysis, one-way ANOVA is used to determine the R square and the P value. In chapter 4, result and discussion, I will divide it into 3 categories, which is the first one is chemical analysis, second is plant growth, and the third one is statistical analysis. As we refer to the graph, coco peat has the highest percentage in moisture content with 82.73% moisture. Similar finding from other studies state that coco peat has extremely high water retention capacity, which resulted in relatively high waterability mixture. A good medium happens when the medium soil have balanced amount of macro and micronutrients. As we compare between these two graphs, we can clearly see that coco peat have both micro and macronutrients required for plant growth compared to the eggshells. Calcium levels are high in both graphs, indicating enough calcium is being supplied to the plant. There is significant difference in potassium levels in coco peat and eggshells. Potassium requirement in medium is important as it is responsible for root development where it involved in water regulations. Although aluminium is not regarded as necessary nutrient for plant growth, its concentration is high in coco peat. Aluminium found to improve plant growth or generate other positive effects only when it presents in low quantities. For soil analysis, red spinach ideal soil pH balance was found between 6 to 7. Results obtained indicate that T1 have pH of 6, which because coco peat has a pH of 5.0 to 6.8, which is neutral to slightly acidic. T2 and T3 have pH of 7 due to inclusion of eggshells. The use of calcium oxide reduces acidity of the soil, releases exchangeable cations, and enhances soil fertility. The graph revealed that red spinach planted in T1 grows larger and healthier plant compared to T2 and T3. T1 plant with height 29.49 cm proved that coco peat medium is the best growing medium for red spinach plantation. This finding clearly demonstrates that the ICP-OES nutrient value are legitimate. The presence of suitable macronutrients such as calcium and potassium in coco peat increases the growth of red spinach, which resulted in taller plants. This is consistent with other research findings that calcium is responsible for enhancing cell growth and development by triggering the growth of the roots. The clear difference of the treatment can be seen on the 6th reading. When the plant reaches its maturity and harvested stage, the presence of available nutrients boosts its development. Nutrients content does affect on the number of leaves. When referred to the graph, the highest number of leaves was in T1 with number of 17.38. The drastic difference was observed on the 6th reading when the plant reached its maturity after 25 days of being planted. Highest concentration of potassium in the coco peat enhanced the build of leaves, and this finding is consistent with other research results. High potassium content improves photosynthesis capacity, reinforce cell tissue, and enable nitrates absorption. Red spinach grown in T1 has the highest value in both height and number of leaves. The plant growth rate was mainly determined by the fresh weight. As we can say from ICP-OES result in coco peat, it demonstrates that coco peat has the greater potassium concentration, which is responsible for the root development and is engaged in water regulation and transfer of plant reserve compound. As the root develops significantly, the uptake of nutrients in the rest of part of the plant also increase and therefore produce a well-growing plant. This is in good accordance with previous results from other researchers, which shows that when the root weight grows, the length and height of the new leaf are increased accordingly. For statistical analysis, p-value obtained for heights, number of leaves, and weight are all less than 0.05. The difference in treatment for the 3 analyses are practically significant. The highest R-square value obtained is in number of leaves, which is 22.4%. In this result, the factor explains that 22.4% of the variation is response. As a conclusion, chemical characterization of cocopit and eggshell as soilless growing medium was successfully performed using FCPOES and also FTIR analysis. It indicates that cocopit consists of calcium, phosphorus, and magnesium as a major nutrient minerals, while eggshell consists almost 99% of calcium as its nutrient content. Red spinach with cocopit treatment demonstrate great growth performance. With sufficient amount of nutrients from cocopit and enough nutrients from the fertilizer, the plant able to grow to the height of 48 cm with 17 leaves. During the observation, the bulk of the plant's leaves were destroyed by insect or parasite. The use of hygropa on the other hand caused leakage and imbalanced distribution of the fertilizer solution. Therefore, for further research, the use of sealed hygropa could be suggested and the use of organic pesticides should be considered. The number of eggshell and its size should be minimized to meet the optimum amount. With all the information that was given, I end my presentation. Thank you.